Sweet, we're live. Screen share. All right. Here's number one.
Okay, so for number one, this is from We Understand. We came up with RNA for the first two because they contain uracil. So there's RNA, double-stranded, and it's circular because it is resistant to the exonucleases but sensitive to the endonucleases. The same with 3 and 4. It is DNA because there is no uracil in it. And then it is single-stranded since it's DNA. And then sensitive to both exonucleases and endonucleases, so it's going to be linear. And then 4 would be circular.
Sweet. And number two, we just kind of go off our notes, and hopefully we have them in our notes. And if we don't have these the answers in our notes or the definitions in our notes, we just go back to the PowerPoint and the lecture notes, and hopefully we can find it in there. And Chris has number 3 on the board.
So for number 3, we worked out the math here, and pretty much to get the length in meters, you just do the 157 million because that's the two base pairs added together for the translocation, and then because each nanometer is 0.34 meters long
Per base pair.
Yeah, per base pair.
There you go.
Anyway, we worked it out, and that's the answer that we got. We also worked this one out for the nucleosomes. No, this is for
It's on B. I got you.
yeah. Yeah, it is nucleosomes.
Yeah, nucleosomes. We just forgot
Yeah.
to label it.
So that's our number for the nucleosomes, and then to figure out the H2A histone for molecules, this was the equation that we used. This is the answer that we got. And then also for D, to figure out the radials, this is the equation that we used. That's the answer that we got.
Nice. And then we got number 4 right here. So number 4, we found the length of our DNA by using our base pairs and the length of each base pair. And then for B, we just figured since the length of DNA is laid out like this way, and then the circumference of that DNA should be the same as the length because it's just in a circle, and we just divide that by pi to get our diameter.
And then for C, the length of the DNA, and then you times that by same 0.34 because it's between each base pair. You get 17,000 nanometers, which then turns into 17 micrometers.
Nice. Here's D. This is how we found out our histones. We took our base pairs, and we weren't sure on if there's 8 histones per nucleus or nucleosome, but we just kind of went with it. We did our base pairs times our nucleosomes per base pair times histones per nucleosome, and we got 80,000 histones. Then we got number 5 on our document.
So for number 5, pretty much each one of these, the nucleosides, nucleotides, ribose, deoxyribose, and pyrimidines, they all have a distinguishing factor in their structure, whether it be like a phosphate group or whether it be a hydrogen on the first carbon instead of oxygen, or instead of OH. So pretty much I looked at the structures online, did some more research, and those were the answers that we got. So
Sounds good.